Instructional video for the spore counting protocol using Terragene's method according to ISO 11138-1 2017. This video uses self-contained biological indicator BioNova BT224 as an example. Remember that specific steps in the protocol may differ depending on the biological indicator presentation, like spore strips, spore ampules, and conventional self-contained biological indicators. For more information, please consult the technical note, Terragene Spore Count Protocol. To ensure accurate results, it is essential to work under sterile conditions to prevent cross-contamination. The materials needed for spore counting are a timer, glass beads, ethanol, sterile water, 1.5 milliliters tubes, 2.0 milliliters tubes, tweezers, sterile tips, vortex mixer, P200 micropipette, and a P1000 micropipette. First, discard the lid and culture medium ampule. With sterile tweezers, remove the filter and place the carrier that contains the spores in a sterile 2.0 milliliters tube adapted to a homogenizer. Discard the biological indicators tube. With a P1000 micropipette, take 600 microliters of sterile deionized water and dispense it into the tube. Now take 400 microliters of ethanol and dispense it into the tube. Close the tube and homogenize the solution in a vortex mixer for 10 seconds. Introduce 6 sterile glass beads into the tube. Close the tube and homogenize the solution in a vortex for another 10 seconds. Set a timer and allow the solution to sit for 60 minutes. After this period, place the tube in a homogenizer with a program set for 60 seconds at 3,600 RPM. Once the program ends, take the tube and shake it gently. Next, carry out the thermal treatment to activate the spores by placing the tube in a water bath at 85 degrees Celsius for the specified duration for each microorganism. 15 minutes for Geobacillus sterothermophilus and 5 minutes for Bacillus atrophaeus, Bacillus subtilis, or Bacillus pumilus. After the thermal treatment, transfer the tube to a cold bath for 15 minutes. After the cold bath is done, take the tube back to the laminar flow hood. Prepare 3 1.5 milliliter tubes, each containing 900 microliters of sterile deionized water. Close the 1.5 milliliters tubes. Take the 2.0 milliliter tube containing the concentrate spore solution and homogenize it in the vortex for 10 seconds. To perform the dilution, take 100 microliters from each tube, dispense it into the next tube, and mix it in a vortex for 10 seconds. Always remember to discard the tip before proceeding to the next tube. Prepare a Petri dish containing 25 milliliters of agarized culture medium, LBA. Then take 100 microliters from the last dilution and place it in the dish. If the biological indicator's initial population is 10 million colony-forming units, an additional dilution must be performed in the previous step, and in that case, the sample taken for the Petri dish must be dilution -4. Now, distribute the sample evenly using glass beads or a Drygalski spatula. When using glass beads, move the plate gently side to side and in various directions until the sample is distributed uniformly. Discard the glass beads. Place the plate in an oven set to the optimal growth temperature for the microorganism. 60 degrees Celsius for Geobacillus sterothermophilus for 48 hours, or 37 degrees Celsius for Bacillus atrophaeus, Bacillus subtilis, or Bacillus pumilus for 24 hours. After the incubation period, remove the plate and count the colonies formed. If the dilution was performed correctly, there should be between 30 and 300 colonies on the plate. To calculate the microorganism concentration, multiply the number of colonies counted by 10 and raise the power of the dilution. As a rule, results should be presented with two significant figures. Therefore, if the count is less than 10, the corresponding dilution is placed in the superscript. If the count is greater than 10, the number is rounded accordingly. According to ISO 11138-1 2017, the spore population must fall within a range of 50 to 300% of the value declared by the manufacturer.